Epigenetic consequences of nutrition in American prisons and its effect on cardiovascular disease. The dots we're trying to connect start with people who are incarcerated, leading to malnutrition from poor diets, leading to nutrients like folic acid and B12 that aren't sufficient, leading to epigenetic changes like methylation that affect different pathways in the ER alpha gene, which affect the endothelium and lead to atherosclerosis. Food in American prisons is decided on by many institutions. The food that inmates are given is governed by state laws, local policies, and court decisions, and these groups' decisions do not always match. As a result, many prisoners are only given. When looking to feed prisoners, prisons look to give them enough food to meet a calorie goal per day, rather than to try and feed them healthier meals. Healthier food often costs prisons too much money, so they do not pay much attention to the quality of food inmates receive. This results in a combination of inmates receiving more processed foods as they tend to be cheaper, and meals that have a caloric value of about 1,800 calories, while the recommended amount is a little more than 2,000 calories and also varies from person to person. This leads to many prisoners becoming malnourished and losing a significant amount of weight very quickly. Poor nutrition is often something that is seen in food deserts across America, along with similar negative effects on health. Nutrition is very important to keep the body functioning how it's supposed to, and poor nutrition can lead to many visible negative effects, such as reduced energy and a weakened immune system, leaving you more susceptible to infection. Poor nutrition can also have a large negative impact on the human genome, causing changes in the expression of certain genes, which leads to the prisoners being at risk for many diseases later on in life, such as atherosclerosis.
Nutrients can play a big role in abnormal DNA methylation. Folic acid and B vitamins are known methyl donors needed for the methylation process to even occur. They donate their methyl group to S-adenosylmethionine, or SAM, which then activates DNA methyltransferase, or DNMT, and presumes ideally normal methylation patterns from there. However, if deficient in these nutrients, many events can happen epigenetically to accelerate atherogenesis. One is that theoretically, if you have a low amount of methyl donors, you will have also a low amount of methylation, which is bad considering normal methylation patterns are needed for regular biological function. But if you have a low amount of methylation, then certain groups that need to be methylated are not getting methylated. Another thing that can happen if you have a deficiency in these methyl donors is you can get a buildup of homocysteine. Homocysteine is usually broken down by folate and B vitamins, and if you follow the pathway on this slide and take the folate path, you can see that folate turns into one of its intermediaries and then reacts with homocysteine to be able to move the methyl group to SAM. SAM then is needed to donate its methyl group to DNMT, and then SAM turns into SAH. This buildup of homocysteine can then cause hyperhomocysteinemia, which can cause vessel damage and also cause a disruption in the SAM-SAH balance. And that is bad because SAH can bind to DNMT and cause hypomethylation events.
In the Hallmarks of Aging paper, chromatin remodeling is defined as DNA and histone modifying enzymes acting in concert with key chromosomal proteins, such as heterochromatin protein 1 alpha, and chromatin remodeling factors, such as polycomb group proteins or the NERD complex. This process is also affected by acetyl-CoA. Acetyl-CoA is known for its main function to deliver acetyl groups to the citric acid cycle to be oxidized for energy production, and this happens through protein, carbohydrate, and lipid metabolism. In this case, acetyl-CoA is the main donor for acetylation of chromatin and proteins involved in DNA transcription and replication. So these levels of acetyl-CoA can increase due to a diet containing high contents of carbohydrates and lipids, because that acetyl-CoA has to work to metabolize that. These elevated levels can lead to change in chromatin structure, suppression of autophagy, which is the recycling and clearing of damaged cells, and also accelerates DNA methylation, which is orchestrated by acetyltransferases. So this kind of leads into that DNA hypomethylation and hypermethylation, which in turn leads to cardiovascular disease. Epigenetic alteration is a change in one's genome by different mechanisms, such as DNA methylation and changes to the chromatin structure. So DNA methylation has two outcomes. There is hypermethylation, which is the addition of a methyl group to the DNA cytosine chain, and this forms 5-methylcytosine 5. On the other hand, there is hypomethylation, which is the removal of a methyl group from the DNA cytosine chain. And as you can see here on this image, it's just a pentene ring, and instead of the hydrogen being a group, it changes it to a methyl. And the other mechanism is changes in chromatin structure, which involves chromatin remodeling factors and protein complexes, and these naturally diminish with age, but this process can be accelerated by poor nutrition and inflammation, which can lead to pathologies earlier in life. Abnormal hypomethylation, which is the removal of a methyl group, has been found in advanced human atherosclerotic lesions, which was found in specifically the aorta and peripheral blood monocytes that were getting excess DNA hypomethylation. And this is from the protein ApoE, which leads into apolipoprotein defects in animal subjects with atherosclerotic plaques. The apolipoprotein E comes from the ApoE gene, and this apolipoprotein E, its function is to combine with fats in the body to form molecules called lipoproteins. And the function of lipoproteins is packaging cholesterol and other fats and carrying them through the bloodstream. So that makes sense in that in animal subjects with atherosclerotic plaques, they have this defect, and with patients with severe vascular disease. When hypermethylation, or the addition of a methyl group onto the DNA cytosine chain, occurs in Kruper-like factors, including KLF2, KLF4, and CREG, can reduce mRNA expression. The function of Kruper-like factors is they're a family of transcription factors, and they regulate multiple types of biological processes that include responses to external stress. So having a poor diet can affect these factors and therefore reduce mRNA expression, and then this in turn causes inflammation and endothelial dysfunction, because endothelial cells is the site of releasing inflammation cells. DNA hypermethylation has been found in the vascular cells of patients with arteriosclerosis, inhibiting KLF2 specifically.
So here is the pathway that pretty much all genes go through to become methylated. DNMT adds a methyl group onto a cytosine molecule. In the cases mentioned above, the high fat and high cholesterol diets have been shown to have an increase in DNMT activity. So more DNMT activity means more methylation. And the pathway between how the diets cause the uptick in DNMT activity is highly debated on and is thought to be extremely complex. Here's a list of genes that have abnormal methylation patterns correlated with diet that have an effect on accelerated atherogenesis and the phenomenon they are associated with. PPAR is abnormally methylated by high-fat diets. PPAR is important for regulating cytokine activity and for upregulating M2 macrophages, which have anti-inflammatory effects, and downregulating M1 macrophages, which have pro-inflammatory effects. High cholesterol diets have shown to methylate the KLF2 and ABCA1 genes. KLF2 is important for homeostasis and protection of blood vessels, while ABCA1 is important for turning LDLs, bad cholesterol, into HDLs, good cholesterol, and moving the HDLs to the liver. Here's the methylation pathway of the KLF2 gene, which is a good reference for the pathways of DNA methylation due to diet. KLF2 can be methylated by high levels of ox-LDLs and free fatty acids. DNMT1 activity is proportional to LDL levels in the blood. When DNMT is activated, methylation at the KLF2 promoter region occurs, and SAM turns into SAH. MECP2, a methylation factor, and EZH2, a histone methyltransferase, then creates a complex. MEF2, a transcription factor that is usually on the KLF2 promoter, is taken off and replaced by the MECP2 EZH2 methylation complex, which turns the euchromatin into heterochromatin and effectively silences KLF2.
The estrogen receptor alpha gene is a gene that binds to estrogen, which is a hormone that's found in males and females that can transcript different genes. It has been found to have protective effects on cardiovascular tissue, and methylation in the CPG island, which is a cytosine and guanine dominant area in the promoter region of the gene, has been correlated with an increase in atherosclerotic plaques. One study actually found that premenopausal women without coronary disease had less active estrogen receptors. So, methylation of the estrogen receptor alpha gene has been found to downregulate that particular gene, which lessens the protective effects it has on cardiovascular tissue, which can lead to dysfunction, abnormal proliferation, and atherosclerotic plaque formation. Atherosclerosis is a cardiovascular disease that's characterized by a buildup of plaque and cholesterol on the arterial walls. Aging contributes to epigenetic changes that already occur naturally, but diet and nutrition also play a very big role in this development as well. The endothelium in arteries is a part of the vascular system, and it's important for maintaining vascular homeostasis and vascular tone, and its dysregulation, if not addressed, can lead to cardiovascular diseases like atherosclerosis. Endothelium dysregulation can be brought on by chromatin epigenetic changes like DNA methylation and histone post-translational modifications that are caused by a poor diet. With high-fat diets in particular, the excess can lead to a buildup of triglycerides and fatty tissues, which then leads lipid levels to increase within our circulation as low-density lipoproteins or LDLs. The LDLs interact with superoxide anions to make oxidized LDLs, which then leads to endothelial dysfunction and cardiovascular diseases like atherosclerosis. This further emphasizes how a healthy diet is necessary in order to mitigate some of the effects that epigenetics already brings.
Diets in American prisons have deficits in many nutritional standards. Poor diet can lead to many morbidities like cardiovascular disease. Diets high in processed fat, carbohydrates, and cholesterol, and low in methyl donors have been shown to be highly associated with abnormal DNA methylation patterns, specifically in KLF2, PPAR, ABC, A1, ER alpha, and many more. These abnormal methylation patterns are linked to atherogenic effects and can be a large factor in the development of atherosclerosis.
There are many outstanding implications associated with these findings. First of all, epigenetic alterations are heritable, meaning they can be passed down from generation to generation. This shows that the effects of incarceration do not end with a sentence, nor do they end with the person who was incarcerated. It could affect their children, both psychologically and physiologically, and it can affect them later in life. This compounded with the fact that Black, Brown, Indigenous people of color are disproportionately represented in prisons means that this is a serious issue for health equity. Epigenetic alterations are reversible, and these cures are currently being manufactured and researched. However, it is highly unlikely that the incarcerated population will be prioritized to receive these treatments. This barely scratches the surface of the consequences incarceration and policing have on our society and public health. Our institutions of justice are more interested in maintaining social order as defined by the wealthy and powerful rather than restoring justice or bringing good health to the victim or offender. Prison health is public health. Once people are freed from prison, they become part of our communities, meaning health issues in the prisons become community health issues and can wear down on our public health infrastructure. Someone and their family's good health should not be threatened by oppressive and biased means of social control. We need to look into healthier and more community-based programs to bring about justice rather than increasing the risk of developing age-related diseases such as atherosclerosis.